everyone. Welcome to today's webinar entitled How to Simplify Your Point of Care Assay Development Process. We're going to give a few minutes for people to join in, a few more minutes. What we can do while waiting for people is for everybody who is already attending, if you can use the chat section and give your name and where you come from, what company you're working for, that would be great. My name is Eric Charrault. I am with Fluxergy, the R&D team of Fluxergy, working on the development of the microfluidic consumable and the different modalities of the platform. I will be your moderator for today's webinar. So again, the title is How to Simplify Your Point of Care Assay Development Process. So it's a joint webinar between Solis BioDyne. We're very happy to have the team of Solis BioDyne with us today and Fluxergy. So the webinar will be going through a few presentations, first by the team of Solis BioDyne, and then I'll be introducing a bit of Fluxergy's vision around point of care testing. And at the end, we will have a Q&A session. So for the question that you might have, please use the chat section and we'll go through them at the end of the presentations. So today we'll be talking, the team of Solis BioDyne will be talking about their temperature stable qPCR reagents and how they were able to integrate them with our Fluxergy open platform. So we have the chance to have with us Dr. Reet Link and Hanna Sein from Solis BioDyne. So I will introduce them quickly. So Dr. Reet is a product development technology specialist who is mainly focused on developing library qPCR and RT qPCR products. She holds a PhD in chemistry from the University of Tartu, where she studied G protein-coupled receptors and reaction kinetics. And we have Hanna Sein, who is a product manager of Solis BioDyne qPCR and RT qPCR product range. She holds a Master in Science in Biomedical Sciences from the University of Tartu. And prior to joining Solis BioDyne, she worked in a research group focusing on understanding gene expression patterns in different model systems of immunodeficiencies. So without further ado, I will leave the floor to Hanna Sein for the onset of this webinar.
Thank you, Eric. Thank you for this kind introduction, and thank you for everyone who has joined us today. And I will just quickly share my screen. So is everything working all right? Good.
Fantastic.
Good, thank you. So we are Solis BioDyne, and I am Hanna Sein. I work here as a product manager. And today we are very lucky that we get to participate in this webinar. So I would suggest we jump right into the first topics. And firstly, I would like to just quickly introduce who we are and what we do. So we are a company located in Estonia, a beautiful and small, currently very snowy and cold Northern European country. And the company was already established in 1995, and we are a manufacturer of stable PCR reagents. We have partners all over the world in more than 110 countries, and we put a lot of focus and efforts into product development and innovation. And last year, we were very lucky to also obtain the award of Estonian Innovator of the Year. And we hold several patents, and we work under strict quality criteria to provide the best products. So what we do, we supply PCR reagents, which can be used to detect whether DNA or RNA to detect the nucleic acids, and whether it's a viral target, bacterial target, human, plant, or animal target. And if you use our products and you need help with your assay development, then we are happy to consult And if the assay has been fully developed, we are also able to offer a large-scale production to ensure a secure supply chain. So what makes our products unique? Many reagents that are used in biotechnology are sensitive to higher temperatures. So, so that makes the transportation and also handling of the reagents really tricky. And the essence of Solis really is our patented technology called the stability tag. And it is a genetic modification that makes the enzymes more stable at higher temperatures. And there, whereas it does not impair the activity, or it even enhances the activity of the enzymes. And it has been incorporated into all of the enzymes and proteins in our product portfolio, so they all carry the stability tag. And the stability is also further supported by unique buffer systems. Why is it beneficial? It reduces risks. Whether the products are stuck in customs, whether you work in high throughput systems where the reagents need to be out of the fridge for a longer period of time, whether there are electricity cutoffs or whatever happens. We can also make more environmentally friendly choices with our packaging, since we only use simple envelopes for transportation. We don't use any styrofoam boxes, and most importantly, we don't use any dry ice. So our carbon footprint is also a lot greener. So our entire portfolio is stable at room temperature for 1 month, and we have many solutions. But today we are focusing on one special enzyme. This is the Solis Fast DNA polymerase. And this has been incorporated into many different enzyme kits and mixes. And why this enzyme? This enzyme has different features like speed, inhibitor tolerance, stability, also good compatibility with UNG systems. And certainly these all features are highly beneficial for anyone doing PCR really. But we do see that there's a certain segment in the market that could benefit from these features the most. And we see that this market segment is the point of care segment. So what is point of care? It's also called near patient testing or bedside testing. And it means that the testing is performed at the site where also the treatment is provided. So it's often performed outside of a classical laboratory setting, away from centralized clinical laboratories. So why is it beneficial? It will give a wider access to healthcare also in remote areas, since in these places often the laboratories might not be so well equipped. So there are possibilities how to do or provide the diagnosis also in these areas. And the basics of it is basically that you get the results fast. There is no time spent on the transportation of the samples to any kind of centralized laboratories, often far away, so where you need to wait for a week or even more to get the results of your tests. So the diagnosis is given faster, and it also enables to start the right treatment from the start, even right when the symptoms occur. So the tests need to be quick and easy to use. And since the tests are given away from centralized, well-established laboratories to other places, then it's also given to the hands of very different professionals. So user convenience is also something that should be emphasized a lot. Solis BioDyne has products that can be beneficial for those assay developers who would like to develop different assays for the point of care testing system. And as was already mentioned before, speed and fast results is probably one of the most important aspects in point of care testing. And the Solis Fast DNA polymerase can enable exactly that, because the extension rates of the Solis Fast DNA polymerase are significantly faster compared to regular Taq polymerase. So fast cycling means also getting the results quicker. The runtime, overall runtime, is of course dependent on what kind of a protocol can you use and also what kind of cycler. So when you use a standard cycling protocol, then this can sometimes take up to 2 hours. And just switching to using quicker cycling protocols, you can already reduce the time by more than 2-fold. And we were also very lucky to find out that when we applied our fast protocol, which is actually not the fastest that we can do, to a Fluxergy platform, we were also able to get really nice runtimes. And my colleague Reet will later also talk about these experiments, so stay tuned for that. So secondly, as was also mentioned, in a point-of-care settings, the testing has been taken from the well-established, well-equipped, well-set-up laboratories to places like your regular office of your healthcare practitioner. And if the setting where the testing is done has not been set up too well, for example, it's a general recommendation with PCR experiments that you have dedicated areas for pre-amplification, sample preparation, amplification itself, etc. But this is always not possible to do in a point-of-care setting. And if routinely the same targets are amplified, then this can lead to carryover contamination and false positive results, which is a big threat to false diagnosis. And how can we combat that? We can combat that by using mixes, PCR mixes that contain the UNG enzyme together with the TUTP nucleotides. And how does that function? What is the mechanism of that? So if the PCR mix has TUTPs instead of TTPs, then these are also incorporated into the amplification product during the reaction. And it's really important to keep in mind how many copies of that amplicon you have in the end. PCR can produce more than millions of copies of a specific target. So these targets, if you don't take maximum precaution, these targets, these amplicons, can easily find their way to also different reactions that you do later from places like the contaminated lab gloves, machines, tabletops, etc. But the solution here is actually that these TUTP containing amplicons are the target for the UNG enzyme. So the UNG enzyme will then degrade these carryover contamination, and later this degraded template cannot be used in the PCR reaction. So in the end, when you get your amplification, you can be confident that what you see is real amplification coming from the real template in your biological sample, and it's not coming from a carryover contamination. In a point-of-care setting, there is often no equipment or time for sample purification, but often sample purification is the first step in all kinds of PCR experiments to get rid of all the other components, so you would end up only with the nucleic acid that you're then going to detect. A solution for that, if you are not able to perform the purification, is to add the biological sample directly to the reaction. However, the problem with that is that biological samples contain many different inhibitors. So it's sometimes difficult to get the same level of sensitivity when doing direct PCR experiments, when adding the sample directly to the reaction. So another way how to then increase the sensitivity is lysed sample, use different lysis buffers, which can then release the nucleic acid into the reaction. Again, the lysis buffers also might come with their own set of inhibitors. So how to then combat this? And one thing to do is to use a PCR reagent that can tolerate these inhibitors. And we have run our own in-house tests, and we have also gathered a lot of good feedback from our good partners who have tested our mixes with different complicated sample types. And we have seen that the Solis fast product range can also work with very complex samples where PCR inhibition is an issue. And here I have also brought out two examples. One of them is sodium chloride, the other one is urea. And why these inhibitors? So sodium chloride is a substance that is present in many different sample preparation materials. For example, buffers like Tris or PBS that are also often used in lysis buffers have a high concentration of sodium chloride. Urea is the most prominent PCR inhibitor in urine samples. And especially in a point-of-care setting, testing urine samples is of high relevance because getting a quick diagnosis for infections like gonorrhea or chlamydia is really important so that the right treatment could be used to stop the infections from spreading. So what we have seen in these experiments is that when we applied a certain concentration of these inhibitors to the reactions, only a moderate decline in performance, just in fluorescence levels, was detected with our mixes. But with sodium chloride, only one of the competitors was able to amplify in these conditions with a significantly higher CT, and no competitor products were able to amplify in the presence of this concentration of urea. So we are really confident that you can test our products also in your direct settings. And last but not least, coming back to the essence of Solis BioDyne, really, the stability. So it's, as was already mentioned before, getting the tests to remote areas where the point of care testing actually take place can be tricky. And our offering of room temperature stable reagents can make it significantly easier. And if the room temperature stable reagents are coupled with lyophilized primers and probes, then it's also possible to ship the entire assay without any cooling. And also, if the testing has been given to the hands of different professionals in different settings, then the ability of being able to set up the reactions without any ice can also be very beneficial. So I have now reached the end of my part. I will now give it over to my colleague Reet. But if you would like to find more information about our products, then feel free to visit our homepage. And now Reet is going to talk about the experiments that we did with the Fluxergy platform, which is also, of course, a point-of-care analyzer with many benefits in that field as well. So we were excited to do these experiments, and now Reet will introduce some results that we got with both our catalog solutions, and also she will introduce a few things that are cooking in R&D currently, so you can stay alert for the new things that are soon coming. So thank you, and Reet, please.
Thank you, Anna.
Thank you. So, can you see the presentation?
Yes, we can.
Perfect. So, hello, my name is Reet Link, and I work as a product development and technology specialist at Solis BioDyne. Solis BioDyne had a great opportunity to test the user experience of the Fluxergy Analyzer and to do a case study on the rapid integration of our robust PCR reagents onto the Fluxergy's open point-of-care microfluidic platform. We used the examples of our probe-based qPCR mixes and RT-qPCR mixes, which are suitable for DNA detection and RNA detection. All these reagents are available in liquid form, but I'm so excited to tell you that we also have lyocompatible and lyophilized reagents in our R&D pipeline, which will be available soon. I will also show in this presentation the results obtained with the lyo formulation on the Fluxergy platform. First, what was our user experience of the Fluxergy Analyzer like? Fluxergy uses disposable PCR cards, which I find labor effective, since they have only a single sample loading port. This is especially useful in case there is only one specimen at a time to handle. The single port is then connected to microfluidic channels, which direct the sample and PCR reagents into 6 reaction wells. This conveniently results in multiple technical replicates. The PCR card is then inserted into the Fluxergy Analyzer, which performs amplification, detection, and analysis of fluorescent signals generated during PCR. The Fluxergy Analyzer has 2 different channels, green and red fluorescent channel. These channels are well separated to avoid any bleeding between them. The Fluxergy Analyzer will automatically generate a report, which makes reading of the results very convenient for the end user. The results are delivered as either positive or negative, depending on whether amplification was detected or not. Also, the analyzer will automatically generate an error text if the test has been failed and needs to be repeated. For example, if the sample needs to be diluted. This is especially beneficial in point-of-care testing, where the analysis needs to be done quickly and easily. In addition, the open platform is user-friendly for those who want to develop their own tests and integrate their own results. Next, let's move to the experiments with our probe-based qPCR mixes on the Fluxergy Analyzer. These mixes are suitable for DNA testing. I will demonstrate the integration of 6 different Solis probe qPCR mixes with or without uracil DNA glycosylase, UNG. These mixes contain DNA polymerase with fast extension rates and strong tolerance against inhibitors, as was already previously introduced by Hanna. These mixes are pipetted together with a primer probe mix, which contains 2 DNA targets and their probes, which are suitable for green and red channels. We use the probe concentrations that were suggested by Fluxergy. As a positive control, we use human genomic DNA. For no template control, we use nucleus-free water, and for contamination control, we used DU-containing amplicons to show the efficiency of UNG for the elimination of the contamination. In these tables, you can see the mixing protocol of reagents. The overall reaction mixture protocol does not differ much from regular PCR. The total volume is 140 microliters, from which 130 microliters is pipetted into the Fluxergy PCR card, which will then be divided into 6 technical replicates within the microfluidic test card, hence the rather large mixture volume. Our mixes are compatible with very fast cycling. This advantage can be utilized if combined with a rapid thermocycler like Fluxergy. In the table below is shown the cycling program that we usually use on Biorad real-time PCRs. This program was transferable without modifications to Fluxergy Analyzer. Compared to the Biorad's fast cycling program, Fluxergy's program was about 20 minutes faster. This allows to save valuable runtime. Our Solis Fast Probe qPCR mix without UNG showed great results: high fluorescence signal, good CT values, and high agreement between technical replicates. According to Fluxergy, a positive result determination with their analyzer requires only 2 positive readouts, which was more than fulfilled with our mix. Also, there was no amplification in no template control. But what happens if the analyses are performed in conditions where there is a high probability with carryover contamination, a frequent culprit when using point-of-care PCR devices? To demonstrate this, we spiked our no template control with DU containing amplicons. Since this Solis Fast mix does not contain UNG, the contamination is not eliminated, and we can see the amplification in the test sample. This would lead to false positive results. But no worries. If there is a problem with carryover contamination, we also have the same mix with UNG. The Solis Fast Probe qPCR mix with UNG showed as good results as the mix without UNG in terms of fluorescent signal, CT values, and the number of technical replicates. In addition, this mix showed no amplification in no template control, even if it was spiked with the same concentration of amplicons that mimic carryover contamination, like in the last experiment. Therefore, if there is a problem with carryover contamination, this mix with UNG would be perfect for eliminating false positive results due to carryover contamination. We wanted to further challenge our Solis Fast Probe qPCR mixes to show how fast we can push the cycling program on Fluxergy Analyzer. For the first perspective of an assay developer, finding the balance between quick runtime and good results need a step-by-step approach, which we have done next. We reduced the amplification time by 3 seconds, and doing that, we won 4 minutes of runtime. Doing so, we did not compromise fluorescence intensity or CT values, although there was a slight decrease in the number of technical replicates. The Fluxergy's requirement of at least 2 positive readouts was more than fulfilled. As a next step, we reduced the amplification time by 5 seconds, which resulted in 5 minutes shorter runtime. Doing so, we did not compromise fluorescence intensity or CT values, although there was a slight decrease in the number of technical replicates. If the amplification time was pushed to the minimal limit, then the runtime was reduced by 6 minutes. Doing so, there was a slight shift in the fluorescence intensity and CT values, as well as in the number of technical replicates. But the requirement of at least 2 positive readouts was fulfilled. These proof of concept experiments demonstrated that our mixes are compatible with very fast cycling, which allows very short runtimes without compromising the results too much. These results could be further optimized by optimizing amplification temperatures and primer mix concentrations. The very fast cycling is especially beneficial for point-of-care testings to produce quick results. Many assay reagents in point-of-care testing systems present a challenge to stabilize. Lyophilization, also called freeze-drying, is often used to achieve the required stability. Lyophilization is a low-temperature water removal process typically used to preserve reagents to maximize the stability and shelf life, maintain function, simplify transportation and storage, simplify assay setup, increase assay robustness, and reduce the risk of sample contamination. In addition, lyophilized reagents can also more easily be added to the different test cards and platforms without the risks of the reagent clogging up the systems, if the reagents need to be stored on the test cards for a longer period. For lyophilization, the liquid PCR mixes need to have a lyo-compatible formulation without components that would inhibit the process, for example, glycerol. Also, the lyo-compatible mixes need some additional excipients that would improve the freeze-drying process, for example, components that would stabilize the mixture or help maintain the structure during freeze-drying. So how does freeze-drying work? Freeze-drying works by freezing the material, then reducing the pressure and adding the heat to allow frozen water in the material to change directly to vapor, sublimate. Freeze-drying can be used to produce either lyo cakes or lyo beads. The names come from a cake-like or a durable bead-like appearance of the freeze-dried material. Usually the quality requirements for an elegant freeze-dried material are no signs of collapse or cracking and product homogeneity. Beads typically have a longer shelf life than traditional cakes because each dose is reconstituted individually. Lyo cakes and lyo beads usually need different excipients and techniques to produce them. In our R&D pipeline, there is also a lyo-compatible version of the Solis Fast Probe qPCR mix with UNG. This mix is in liquid form and ready to be lyophilized to produce lyo cakes without the addition of excipients. The liquid lyo-compatible mix retains similar properties as the original Solis Fast Probe qPCR mix with UNG, as demonstrated by the fluorescence intensities, CT values, and number of replicates, and by the efficiency of UNG for eliminating carryover contamination if the test sample was spiked with applicants that mimicked carryover contamination. Since Solis BioDyne has lyophilization capabilities, there is also a lyophilized version of the Solis Fast Probe qPCR mix with UNG in our R&D pipeline. As shown on the photo, we have produced lyo cakes of the mix with elegant appearance. The lyophilized mix retains its great properties as shown on the positive control and by the contamination control of the lyo beads. In addition, we have another lyo-compatible version of Solis Fast Probe qPCR mix with UNG, which formulation is suitable for those users who want to add their own excipients to produce either lyo cakes or lyo beads. As shown on the photo, our first batch of the lyo beads have been successfully produced. The product characterization and stability tests of the beads are ongoing. Finally, let's move to the experiments with our probe-based RT-qPCR kits on the Fluxergy Analyzer. These mixes are suitable for RNA targets. In our R&D pipeline, there is also a lyo-compatible version of the one-step RT-qPCR direct kit, which includes lyophilization excipients to produce lyo cakes. This mix is also suitable for those users who want to add their own excipients to produce lyo beads. The probe-based RT-qPCR kit components are pipetted together with 4-plex primer probe mix, which contains 4 different RNA targets and probes that we usually use with our SARS-CoV-2 RT-qPCR kit. Although Fluxergy Analyzer has 2 different detection channels, and all 4 targets could not be detected in this case, we wanted to show that even without optimization, multiplex assay kits are compatible with Fluxergy Analyzer. As a positive control, we use synthetic SARS-CoV-2 RNA. For no template control, we use nucleus-free water. And in the table, you can see mixing protocol of reagents. The total volume is 140 microliters, from which 130 microliters is pipetted into the Fluxergy PCR card. For the cycling program, we used a similar program that Fluxergy uses for the amplification of SARS-CoV-2 RNA on their platform. Our one-step RT-qPCR direct kit showed great results, even though our primer and probe mix was not optimized for the Fluxergy Analyzer. Also, there was no amplification in no template control. To optimize the results, we optimized our primer and probe mix based on Fluxergy's suggestions. We used 2 different SARS-CoV-2 RNA targets, but their probes were both labeled with the same dye, FUM dye, so they could be detected in the same channel. Also, we increased the concentration of primers and probes. In addition, we changed the cycling program slightly to be more similar with Fluxergy's SARS-CoV-2 program. In this case, the fluorescence intensity values were 2 times higher, and the CT values were shifted earlier. Also, there was no amplification in no template control. So there is a possibility to use off-the-shelf kits that are not developed for the Fluxergy Analyzer, but on the point of view of assay developers, we suggest optimizing the assay design to improve the results. In summary, what was our experience with Fluxergy? Fluxergy Analyzer has a small and practical design. It has 2 channels, which provides an easy framework for assay design. The platform is compatible with the different cycling protocols and enables very fast cycling, very fast results. The interpretation of results are easy. Their platform has a good compatibility with our Solis BioDyne reagents. And last but not least, they have a very supportive team. And if any of our products caught your interest, then we would be happy to send you a free sample, which you can also try out in your test systems. Feel free to send your request to info@solisbiodyne.com. A good news is also that we have a discount code for webinar participants, and the code is webinar20, which you can use when ordering to get 20% off your order. So thank you from my side, and I would give it over to Fluxergy next.
Thank you, Dr. Reet. Yeah, it's been, I think, very good on our side as well to be collaborating with you on this project. And it's quite exciting to see all those preliminary results that you got, and very looking forward to the next steps. I will go ahead and talk a bit more about Fluxergy. Fluxergy is a medical device manufacturing company. We are located in Irvine, California. We're about 100 people right now, and we are active in the human and veterinarian space, mainly equine. We have a few CE-marked products. And over the last few years, we've been focusing on developing our manufacturing capabilities as well as our quality system. So we have now a beautiful 70,000 square feet manufacturing facility, which allows us to produce around 10,000 test kits per month. And we have 100% traceability on the product, 100% inspection on the product as well. And I guess our goal is to disrupt the point of care testing with our product, which is around a multiplex, multimodality, open platform system. So there is, like, with one single device, you'll be able to do a lot of different type of testing modalities, such as molecular testing, immunochemistry, chemistry, and cytometry. It's an open platform concept, so meaning any assay developer can come and develop their technology for the platform and bring their assay to the point of care. So when we look at the point of care landscape, like we know that point of care testing will be part of the future of diagnostics testing. I think the pandemic really highlighted that a combination between a conventional external lab setting with a point of care testing is really able to handle properly any type of public health related issues. As was discussed earlier by Hanna, there's also a lot of different aspects of point of care. When we talk about low income countries, for example, they don't have necessarily any proper infrastructure to have a proper lab. So point of care definitely has a lot of benefits in those type of settings. More general, when we talk about emergency, critical care, even primary care, there's definitely a lot of benefits around, I guess, timeliness of the diagnosis, and also improving the patient outcome because of this timeliness. So all the point of care devices, I guess, are now towards getting always like smaller, faster results, cheaper and more accessible. One challenge that remains, though, is that all those devices are really specific and only looking at a specific type of tests. When we look at having, for example, the complete overview of a patient diagnostics, we need more than one type of tests, meaning that for the point of care setting, there will be a need to acquire a different type of point of care devices, meaning require more space, which we don't necessarily have. There will be a lot of, I guess, more upfront costs because we need to purchase those different devices, and also more overhead costs with respect to the training of the staff on using those devices and also maintenance of the devices. What's also very fascinating is now in this, I guess, post-COVID era, everybody is aware of rapid testing and point of care testing, meaning that there will be soon the need to increase the test menu offered at the point of care. And so this is where, I guess, Fluxergy's vision is around the point of care testing. With our multiplex, multimodality open platform system, really you have the possibility to have one single device and be able to do a lot of different type of tests. So like I said earlier, nucleic acid amplification, immunochemistry tests, clinical chemistry, and cytometry. And since it's an open platform system, there will be the opportunity for a developer to bring their own tech and own assay to the point of care, meaning that represents a huge opportunity to have an ever-growing test menu that is offered. The whole system is designed for the point of care, so all the different workflows associated with the different type of modalities are ready for the point of care. So they're very low complexity. All the tests are simple to answer between 15 and 60 minutes. The longest time are for the PCR tests with the different cyclings. And our microfluidic consumables is very cost effective. And so this is something also related to one of the core value of Fluxergy, which is around manufacturing and scalability. So any of the R&D teams at Fluxergy, when we look at designing and developing a new product, there's a couple of fundamental principles that we try to keep in mind. They're mainly around the flexibility and modularity of the product. And this is directly transferable to our manufacturing and automation processes, which allow us to be really cost effective, even at very low volume. So from, very cost effective at low volume, and also giving us an opportunity to be more scalable. So at the moment, we can do around 100,000 test kits per month, but there's a straightforward path towards 1 million test kits per month. So if we look at the PCR technology capability of the platform, as was presented by the team at Solis, the analyzer and the consumable are able to handle temperature control. So all of the main PCR types are available to be performed on the device. So we're talking about multiplex PCR, RT-PCR, isothermal PCR. We do have 6 individually controlled reaction chambers. And we do have a couple of fluorescent channels, one in the cyan, one in the amber, meaning that we can do a colored and spatially resolved multiplexing PCR, which gives an opportunity for us developers to tune down the complexity of the mixes. The latest addition to our PCR family is a multiplex PCR test card that includes lyophilized reagents, meaning that it's even more ready for the point of care. And as was mentioned from Solis and Dr. Reet, lyophilized reagents really have the advantage of we don't require the need of a cold chain anymore. And this is done without adding any complexity to the workflow. So really for the end user, it's just about collecting the sample, diluting it in some kind of buffer solution, and introducing it in the test card. And then all the other steps are performed on the test card during the test. So we're talking about rehydration of the reagents, mixing of the reagents, and all the different thermolysis steps and PCR cycling. For the assay developer, what it means is it really doesn't really change the workflow from a typical conventional PCR thermal cycler. There are some restrictions in terms of sample volume. So we need around 100 microliters in the test cards, which correspond to a sample volume of around 10 microliters. And we have the fluorescence channels that are defined by the hardware. Other than that, you will have access to full control over the different parameters of the testing. So for example, for PCR, you will have control over optimization of the temperature set points, number of cycles, and duration of the cycles, and really go through a typical assay development as you would do on a regular thermal cycler. The test card consumable that we will be giving to assay developers is really the final product. So whatever you'll be developing on will be the final product. And I will conclude with this slide showing, I guess, the whole concept of the open platform, I believe, is also addressing another challenge that is not in the point of care setting, I guess, markets. It's very hard and complicated to design, develop, and manufacture your own device. And so the opportunity that we offer to us developers is, well, you don't have to worry about all these orange boxes which represent all the work related to the hardware side. You just have to focus on developing the assay and have it validated and optimized for the Fluxergy platform. And it represents a very straightforward way towards commercialization without having the need to worry about any regulatory aspect about the device and all the cost and time effort related to that. So I will leave it here. And thank you everybody for the time and the attention. And now we'll be able to go to answering some questions. So, I'm sorry for earlier. I missed if we went through the question already or not. We have a couple of other questions here. We have a question around the production of large quantities of Lyo beads. So the question is, can you produce large quantities of Lyo beads, 50,000 batch size? So I believe this is for the Solis team.
Yes, thank you. Thank you for this question, and thank you for the interest. We are currently still working on our solutions for lyophilization. So these are still in the test product phase, and we have produced our first batch of the Lyo beads, but we would love to hear more about your interest. So feel free to send us an email to info@solisbiodyne.com, and we will make sure that you get the information when we know more.
All right, another question here is about the preparation of the nucleic acid amplification. Could you explain a bit more whether the nucleic acid amplification is done separately before loading the qPCR mix with the sample to the Fluxergy test card?
I think this is a question to Reet.
The PCR reagents and the test sample are loaded onto the PCR card and then inserted into the Fluxergy Analyzer, and the Fluxergy Analyzer performs the amplification then. Hopefully I answered the question.
Yeah, I guess for the Fluxergy test card, so depending on the workflow, so I believe the workflow that you've been using was for the liquid reagents. So in that case, there is the need to prepare the whole reaction master mix outside the Fluxergy platform, and then introduce the whole mix into the test card. And like I mentioned, we have a new multiplex test card with lyophilized beads, and so this is really removing the need to mix the reaction mix prior to introduction of the test card. So it's really sample collection, dilution of the sample, and introduction in test card. So hopefully a more point of care ready workflow. We have another question here. Do you see the use of direct qPCR reagents as becoming more frequent in the laboratory in comparison to using extraction-based kits?
Yes, from our side, we definitely are getting more and more requests regarding inhibitor tolerance and the capability of using our reagents in direct settings and skipping the extraction phase. And based on the interest that we get from our partners and from the market, we can definitely say that the need for inhibitor tolerant master mixes is increasing, and more tests are performed in a direct settings. And I guess it also makes a lot of sense, because it will save a lot of time, a lot of time and money if you're able to skip that step. So I would assume we see that the interest is only growing in the near future.
All right. We have another question here around the type of sample. Have you tried more changing samples while testing your assays? Sample like poultry samples, feces, wastewater.
Yes, actually our many partners have reported many interesting applications where they have used our products with more complex samples, using them either directly in the detection or using some kind of a simplified sample preparation step before the PCR. So regarding wastewater, yes, our products are also used in the wastewater screening for SARS-CoV-2 virus. So we have we have gathered good feedback from a partner who is doing that. Regarding other complex samples, one tricky sample type is always blood. And we have had some good feedback from partners who have used dried blood spots and used just a simple lysis buffer and using then this simply prepared sample directly in the qPCR, and also getting satisfactory results with the blood samples. Also different kind of plant samples like seeds and leaves and also soil. We have had many partners report that they found our products working really well with these sample types as well. So we can only encourage also others to who are struggling with their inhibited samples to perhaps try out ours, and maybe this can be the help that's needed. Yeah.
Thank you. I have a question here for Fluxergy, also on the sample type. What type of sample have been directly used on the Fluxergy cartridge? So on our side, I guess the main sample type that we're using are nasopharyngeal swabs samples. We also have blood, capillary blood, and we've been also working with urine samples. And yeah, in R&D, we've been working with all kind of other type of samples, mainly blood, nasopharyngeal swabs are the main ones now. We also have a environmental swab for our salmonella assay for PCR tests. These are the main samples that we've been using. All right. How are we doing on time? I think we're very close to the end. So we're going to answer, we're going to reply to some of the other questions and try to forward them to people who ask them. We will leave it here for today. I'd like to thank you, everybody, again for attending and for your interest. And thank you, the Solis team, so Hanna and Reet, thank you very much for the presentation. And like I said, really looking forward to the next steps. And I wish you all a great day.
Thank you.
Bye-bye.
Thank you.